Okay, this next lab is called Fermentation of Carbohydrates. And what you've been learning about, I believe it's in chapter, I want to say 5 or 6 of the textbook, talks about metabolism of microbes. And we know that breaking things down is called catabolism and building them together is called anabolism. In fermentation, recall that there's aerobic cellular respiration and then there's anaerobic cellular respiration. And lots of microbes, if they start to run out of oxygen, with or without oxygen, they may go through a fermentation cycle. So instead of going through all the steps of glycolysis, the Krebs cycle, and then the electron transport chain, they will only go through glycolysis. And then that pyruvic acid that's made at the end of glycolysis will be used to control the hydrogen ion concentration and reduce the hydrogens in the end rather than oxygen at the end of aerobic cellular respiration. And so if they go through a fermentation pathway, they're going to produce different kinds of acids. Okay, so that's what we're going to be looking at today, is whether or not they produce an acid through fermentation, or maybe they go through a different pathway and they produce more of a neutral product. And so based on that, we're going to see some color changes. So this is kind of cool when you see the different color changes. Although your slides were very pretty last week, all the different colors. All right, one thing I want to add is this procedure starts on page 93. And it has the fermentation tubes and the MRVP tubes. I'm going to add one tube that was in a previous edition of the book, and I like to use it now because we're going to use it later, so it won't be sneaking up on you later in the semester. The last tube that we're going to use is called the Simmons citrate tube. I'll get to the tubes in just a minute. But what I'd like for you to do is refer over to page 107 of your book. On that, it talks about rapid ID methods, and what I'd like for you to do is go down to, there's a major paragraph and then there's 4 little paragraphs for I, M, V, and C. I want you to look at the C portion. That stands for citrate, okay? And when you read that, you'll realize that if a microbe uses carbon as its source of energy, it's going to turn the Simmons citrate tube from green to blue. So this is the Simmons citrate tube. You notice that it's an agar slant, and it starts out green. So if that bacteria is positive for citrate, then it's going to turn it blue. Okay, so that's going to be an addition. So you'll have to make a note in your lab manual back on page 97 where we're starting out, that or 93 where we're starting out, that add the Simmons citrate tube. Okay, so now for this particular experiment, just like the last one we just did, we're going to have lots of tubes. In this case, we have 3 of what we call fermentation tubes. Then we have 4 MRVP tubes, and then we're going to add the Simmons citrate. So far we've got 8. And then you're going to have at least 2 cultures, if not 3, and it depends on how they're assigned. So turning to page 95, okay, there's a list of cultures that are going to be used. E. coli, Enterobacter rogenes, Acarigines faecalis, and Proteus vulgaris. For the first part, which is the fermentation part, so the procedure below that says procedure fermentation tubes, I am going to assign you particular cultures to use. So I'll just go group by group, and I'll give every group 2 cultures, okay? And for that particular one, you're going to, wait a minute, I'll probably give you 1. 1 or 2, I haven't decided yet. So anyway, if I give you 1 culture, you'll end up with 3 of these tubes. If I give you 2 cultures, you'll end up with 6, okay? And it'll probably be limited when we get our supplies, however many tubes we have there. If I have enough to assign 2 per group, I'll do 2, but if we don't, we'll just do 1, okay? So regardless, you're still going to do the same procedure, all right? So pretend that I've assigned you E. coli, okay? So you'll need E. coli for the fermentation part, the fermentation tubes. Now, you'll notice there's a second procedure called MRVP test. And for that procedure, it says inoculate 2 MRVP tubes with E. coli and 2 with Enterobacter. So that's why you have 4 tubes. You're going to be putting E. coli in 2 of the tubes and Enterobacter in the other 2 tubes. Now do note that E. coli and Enterobacter were also possibly assigned to you in the fermentation tube. So you may or may not use the same ones, just depending on what culture you were assigned in the first part. Okay, so you may end up with one culture, 2 or 3, depending on which ones you're assigned. Okay, not 1, but 2 or 3. All right, so the first one that we're going to do is the fermentation tubes. What I want you to notice is these fermentation tubes. You might see bigger or smaller ones, and they're going to be a shade of red. I like for them to be red, but sometimes they end up a little on the orangey side or the peach side. Either way, we want them to start out red, because the indicator in here, the pH indicator, phenol red, is what's going to turn yellow in the presence of acid. So if your bacteria go through fermentation and they produce acid, your media will turn yellow. Okay? The other thing is sometimes when they ferment, they also produce gas. Okay? So if you'll notice, I don't know if you can or not, I hear it clicking, but inside this tube, there's a really small tube that's upside down. And that small tube that's upside down is called a Durham tube. And this one is completely full. This one here has an air bubble in the top of it. So when detecting the presence of gas, you're looking for an air bubble. Okay, so this one, since it didn't have a bubble, the next time when you go to look at it after it's been inoculated or sitting in the incubator for a week, you might notice a bubble in there, and then you would note that it produced gas. This one already has a bubble in it, so you'd have to make a note of the size of that bubble, so that when you go to look at it the next time, you'll have to note if it increased in size, the bubble itself. Okay? So for these tubes, we're looking for 2 things. Did they turn yellow and produce acid, or did they produce a gas bubble? Okay? Now, for inoculating these tubes, we're just going to use a loop. Okay, so we would sterilize it like we've been doing. Now, this is the first time that we've actually used tubes that have screw-on lids. Okay, these are the easiest to work with, but sometimes we get them with screw-on lids, so they're a little bit harder to open and close. Okay, now you're going to put the bacteria that you were assigned into all 3 of the tubes. Okay, the different sugars will be lactose, sucrose, and glucose. Okay, so we have lactose, sucrose, and glucose. Okay, so we're going to put our bacteria into all 3 of those. So I'm going to take the first 2, and just make sure you have, before you do any of this, you would have labeled it with your name and the name of the bacteria. Don't forget the labeling part. I'm going to loosen that a little bit before I start grabbing with my fingers. So I've already sterilized my loop and it's cooled down. So I'm going to take my lids off. I'm going to go into my culture like I've always done, and then go into the new one. Okay, sterilize my loop. Now, one note I want to make is on these screw top lids, you don't want to close them completely tight, because if you do, you're cutting off the bacteria's oxygen supply. And at this point in the lab, unless you're told differently, you want your bacteria to have some oxygen. So what you can do is you can close it to where it closes, not real tight, but it's completely closed, and then give it a turn opening. You don't want it falling off, but you want it to be loose. Okay? We would use that same procedure and put the same bacteria into your other 2 tubes. Okay? Now, the Simmons citrate one that I had mentioned, remember that's an addition, so you're going to kind of write it into your book. The same thing, whatever bacteria you were assigned, you would put into the Simmons citrate tube. Now, recall that this, and I'm just going to pretend that I've done everything else, and I just want to show you again how to inoculate a slant. Okay, remember I already have, pretend I've already sterilized everything, I already have the bacteria on the loop, and you don't want to gouge the auger, you just want to start at the bottom and slowly come toward the top. Okay, and you would do all the sterilization that we've done before. Okay, so pretend I have all of those inoculated, I've flamed my loop and done all those things. Now, going to the MRVP test. Once again, here are the 4 MRVP tubes. You're going to put E. coli in 2 of them, put Enterobacter in 2 of them. Okay, and there's a reason for that, okay, because in the second part you'll need 4 tubes. So we're going to do the same procedure. I'm not going to open these up. I don't want to keep wasting media, but once again, You're holding it with 2 hands. Loosen this one first, and then after you've sterilized your loop, you're going to pull off the 2 lids, go from here into there. So we're going to put E. coli into 2 of them and put Enterobacter into the other 2. Okay, so for those procedures, that's all you're going to be doing today. And then the next week, you will finish these labs and then start a new one. So that's going to be kind of the procedure from here on out.